Hello, students. This is Dr. Pratibha K.S., Head Department of Biotechnology at KLE Society's S. Njengappa College, Rajayanagar, Bengaluru. Today, I'm before you to present Biotechnology syllabus of SEP Paper 1, Cell Biology and Genetics, and our S. Njengappa College affiliated to Bengaluru City University. All right. And today we are going to choice-based credit system as per SEP 2024 for the academic year 24 and 25. Okay, let us go through the syllabus. 1. Cell biology and genetics. Here you can go through the regulations and syllabus for biotechnology. Eligibility: only those candidates who have passed a pre-university course or an equivalent course with biology as one of the optional subject are eligible to take this biotechnology course. And here you can see the scheme of instruction or maybe the examination, the different rules and regulations. You can go through this. Okay. Now let us go through the scheme of instructions and credits. As this semester, you are studying cell biology and genetics, theory paper code BTT 101. And per week, theory practicals will be 4. And duration of the exam for this theory, 3 hours. Okay. And the class IA marks will be 20. And you're writing the theory examination for 80 marks. So total 20 marks IA and 80 marks your theory paper, together 100 marks. And this gives you 3 credits. Then practical BTP, Biotechnology Practical, BTP 102, paper title, Cell Biology and Genetics, practical. In a week, 3 hours of one practical you need to attend. Then duration of the exam for your practical 3 hours. Then IA marks 10 marks. You're writing the practical paper for 40 marks. So 10 IA marks and 40 practical paper, together it's 50 marks. Okay. Then credits for practical is 2. And here you can see the paper 2, that is second semester paper, microbiological methods theory and respective practical. We'll go through this in the next semester. Okay. Yeah. Please make a note, and or maybe you can take a screenshot. If you make a note in your notebook, that will be great, because once whatever you are listening, once you made a note, that will be good for you too, so that you can turn the pages and can go through. And this is your theory scheme of examination pattern of biotechnology paper 1. Duration 3 hours, maximum marks 80. These are the 3 patterns. First main, answer any 10 of the following. Out of 12 questions, you need to answer any 10, and each question carries 2 marks. So this will give you 20 marks. Okay. Then second main, answer any 6 of the following out of 8. 2 will be the option. So for 6 questions, it carries 5 marks. So it's 30 marks. Then third main, answer any 3 of the following. Out of 5 questions, you need to answer 3 marks. So this is 3 into 10. This is not 5. 3 into 10, that is 30 marks. So 30 plus 30, 60. 60 plus 20, 80 marks. So your theory examination for 80 marks, and 20 marks will be your internal assessment marks. I hope you're able to understand, right? Now let us go through some of the instructions related to the theory paper. Course title BTT 101, Cell Biology and Genetics. Okay, total contract hours is 56, and credit will be here is 4 credits. Okay, as I told earlier, 20 is the IA marks, and duration of the exam will be for 3 marks. And writing part, theory examination, 80 marks. Now let us go through the BSc Biotechnology first semester paper 1, paper code BDT 101, Cell Biology and Genetics. Okay. Unit 1, Introduction, Cell and Cellular Organ. This Unit 1 carries 14 hours. Okay. This unit carries 14 hours. It means within the 14 hours, we should be able to complete this particular unit. Okay. Let us go through the Unit 1, Introduction, Applications and Scope of Biotechnology. Then Cell, historical perspectives, discovery of cell, the cell theory, ultrastructure of a eukaryotic cell, both plant and animal cells. Okay, here you are studying only the eukaryotic cell, both plant and animal cells. So not nice to focus on prokaryotic cell, but you need to understand the concept. Prokaryotic cell, that will come in the next semester, that is second semester in microbiology paper. All right. I hope you're able to make a note. Okay. Otherwise, you can pause the video or you can take screenshot and write the syllabus in your theory notebook first page. First page, there should be compulsory theory syllabus, written theory syllabus. Okay. Then structural organization and functions of cell wall and plasma membrane. Then structure and functions of cell organelles like cytosol, endoplasmic reticulum, Golgi complex, mitochondria, chloroplast, ribosomes, lysosomes, nucleus, nucleolus, vacuole, cytoskeletal structure, microtubules, microfilaments, and intermediate filaments, which comes under cytoskeletal structure. So this is all about the unit 1, right? Now let us go to the unit 2, that is chromosomes and cell division. Again, this unit 2 also carries 14 hours. General introduction, discovery, morphology, and structural organization of the chromosome, specifically metaphase chromosome, you need to study here. Then further about centromere, secondary constriction, telomere, chromonema, euchromatin and heterochromatin, and chemical composition. Further, classification based on centromere. Okay, then karyotype definition, ultrastructure of chromosome, that is specifically nucleosome model. Okay, then let us go to the special types of chromosomes, salivary gland chromosomes and Lamprecht chromosomes. Okay, then cell cycle, phases of cell division, mitosis and meiosis, cell senescence and programmed cell death, structural numerical chromosomal aberration. So all these which comes under the unit 2, that is chromosomes and cell division. Okay, I hope you are making note. Now let us go to unit 3, inheritance and gene interaction. Here you are going to study history of genetics, Mendelian theory, law of inheritance, dominance, segregation, incomplete dominance, codominance with an example. Okay. Then law of independent assortment, test cross and back cross. Then you are studying about maternal inheritance. Examples like plastid inheritance in Mirabilis jalapa, kappa particles in paramecium, and peptide characteristics in yeast. Then sex-linked inheritance. Okay. Then the next concept is gene interaction. Supplementary factors, example, Coombe pattern in fold. There are so many other examples will be there, but in the exam, you need to focus only on this one, Coombe pattern in folds. Then complementary genes, flower color in sweet peas, epistasis, plumage color in poultry, then multiple allelism, blood groups in human beings. So all this which comes under the unit 3. Next, unit 4, linkage and mutations. Under this linkage, general introduction, coupling and repulsion hypothesis, linkage in maize and Drosophila, mechanism of crossing over and its importance. Okay. In chromosome mapping, you are studying only the linkage map in maize. Then coming to the mutations, here you are going to study introduction, types, spontaneous and induced mutations, then mutagens, physical and chemical mutagens, mutations at the molecular level. Next concept, sex determination. In this concept of allosomes and autosomes, sex determination in plants and animals like XXXY, XXXO, ZW, ZZ, ZO, ZZ types. Okay. Then chromosomal syndromes. Chromosomal syndromes in man like allosomal and autosomal conditions. Allosomal like Klinefelter syndrome and Turner syndrome, whereas autosomal, Down syndrome and Creutzfeldt syndrome conditions. All this you are going to study under unit 4, linkage and mutations. Okay. I hope you understood all the 4 units of paper 1, biotechnology syllabus. First one, we went through unit 1, introduction, cell and cellular organelles. Then unit 2, chromosomes and cell division. Unit 3, inheritance and gene interaction. Unit 4, linkage and mutations. Okay. Now, this is all about your internal assessment marks for 20 marks. House examination or maybe a test carries 10 marks. Written assignment or presentation project or any kind of term projects, it carries 5 marks. Class performance or participation carries 5 marks. So total in theory, you have 20 IA marks. Okay. Then these are the experiments you are studying in your biotechnology practical class. For the biotechnology practical class, you need to keep one separate 50-page notebook only for the practical experiment. And you need to call that as the observation book for biotechnology paper 1. Okay, experiment number 1, operation and working principle of simple and compound microscope. Second experiment, study of micrometry, measurement of onion epidermal cells and yeast. Experiment number 3, study of mitosis in onion root tips, then preparation of temporary squash. Fourth experiment, study of meiosis in grasshopper testes, onion, Rio flower buds, and preparation of temporary squash. So to study this meiosis stage, we can go for the sample like grasshopper testes, or maybe onion sample, or maybe Rio flower. Okay, onion flower bud also you can use. Then experiment number 5, preparation of buccal epithelial smear. Okay, to observe the Bohr bodies. Then experiment number 6, isolation and staining of mitochondria or chloroplast. 7th one, karyotype analysis, human both normal and abnormal you are studying. Abnormal like Klinefelter syndrome, Turner syndrome, Credit syndrome, okay, Down syndrome. Like that you are studying under the karyotype analysis. And also you're going to study the onion karyotypes. Last experiment, simple genetic problems on the interaction of genes. Based on the interaction of the genes, you are going to solve the problem. So this is all about the 8 experiments of the paper 1, cell biology and genetics. Okay. Okay, now the total practical IA marks is 10. There we are going to distribute like house examination, maybe a test, carries 5 marks, class performance or participation, 5 marks. This 10 marks under your practical. Then coming to the biotechnology first semester practical BTP 101 cell biology and genetics practical examination paper or maybe scheme of examination paper. I've already showed you in the beginning theory examination paper pattern like that. This is the practical examination paper pattern. Duration 3 hours, maximum marks 40 hours. Okay. Question number 1, measure the size of the given material A using micrometer. It carries 8 marks. Prepare temporary squash of given material B for mitosis or meiosis and comment on the observed stage. This carries 10 marks. Prepare a buccal epithelial smear and report the observation carries 4 marks. Identify and comment on the karyotype C and D. Each karyotype carries 4 marks. So 8 marks. Solve the genetic problems. It carries 5 marks. Then coming to the class record, it carries 5 marks. Okay. Here in the question number 1, micrometer when you are performing for the calculation part. Okay. All those things 4 marks. Writing pattern. Then the performance is 4 marks. So total 8 marks. Then while preparing the mitosis or maybe a meiosis slide, performance carries 5 marks. Writing part carries 5 marks. Okay. And here in the buccal epithelial smear, performance carries again 2 marks. Writing part 2 marks. So total 4 marks. Identify and comment on the karyotype. What you identify that carries 1 mark and what writing pattern carries 3 marks. So total 2 karyotypes each carry 4 marks. Then any one genetic problem will be given. You need to solve. So kindly make a note of all these question paper pattern of the practical in your observation notebook so that you can focus on those particular experiments. Then while studying this first semester biotechnology paper, we do give study material to study. Okay, we can go through that one by going through the study material. You can score 95%, but still you need to visit a library and you can make your own notes by referring foreign author book or Indian author book or maybe a local author books. Okay, and these are the list of the books. If you want, you can purchase. Otherwise, you can refer all these books in the library. Okay, you can take a screenshot of this particular textbooks and reference books list. I hope you understood. At the end of the class, you should not be like this. Okay. If you're like this, what happens? Then totally waste of time. That's why please focus. And I like active students, active learner, very, very important in the classroom. Okay. I hope you're not like this. I hope everyone are happy. Best of luck. Stay blessed. And you be the gift to the world. Okay. And one more homework is there you need to do in your free time. Take one notebook or maybe a diary. Okay. Separately keep that one only for writing your future consequences or maybe your dreams, all those things. Okay. Wow. First point, write your 10th marks. Okay. Then percentage of first PU and second PU mark. Then write your expected or target marks at degree level. Now you are freshly joined to the first semester. So write your expectation. Okay. How much are you expecting in this particular first semester? Okay. Dream big. If you make your own target, then you can go behind that. If there is no target, nothing means then you will be somehow you are going to lead your life. That's why put some targets so that will be useful for you. Then second one, write your future plan or dream after 3 years. So after 3 years, what do you want to do or what is your aim? Please write around 3, 4 dreams you can write. Write your future plan or dream after 5 years. After 5 years, what do you want to become? Or if you have any kind of any dreams, please write down those dreams, okay? And don't dream only a 1, 2 dreams, okay? Go for on more than 5 dreams because you need to go behind that. If one dream you are unable to succeed, at least you can focus on other dreams. Write your strength, challenges and weakness. To write all these questions immediately, do not write think. Please take 2 days, 3 days, 4 days, 5 days. Then please write the answers for all these questions. Write your happy moments spent in your life. Okay. Genuinely, you're supposed to write the answer for all these questions. Right. Last time, when did you laugh very strongly? Okay. Think twice, thrice. Then you write the answers. Write how you want to help your parents in future. Then eighth one, write how you want to contribute yourself to the society. Okay, you should have some your own strategy to contribute yourself to the society in different manners. You need to think about that. In your view, what is life? Okay, then who are you? You need to question yourself and you need to answer for this. Then your life goal in your life, what do you want to become? What is your life goal? All those you need to think and write in your notebook so that you are going to be a very good citizen in the society. I hope I made myself crystal clear. Please accept all these as a challenge and go through that. Okay, show must go on. Like you, and something goes wrong, also you should also learn the concept like move on. Okay, don't hang over on that only negative incident, whatever has happened. Just move on and think positively. Okay, I hope you enjoyed today's session. I, Dr. Pratibha, signing for the day. Thank you, everyone, for being with me. Take care. Bye bye.